A very good afternoon to one and all. On behalf of St. John Institute of Pharmacy and Research, I, Rennie Daniel, student of Masters in Pharmacy in Quality Assurance, extend a warm welcome to each one of you to our 23rd session of SCIENTIA. Before commencing the session, let's have a small video of our college campus. Now, I would like to commence with our current session, which is entitled Malarial Vaccine. It is a pleasure for me to introduce our eminent speaker for today's session, Mrs. Ekta Thakor ma'am. Mrs. Ekta Thakor ma'am has been working as an assistant professor in Department of Pharmaceutical Chemistry at SGIPR since 2019. She has completed her B Pharm and M Pharm from the University of Mumbai. She has 5 years of academic experience. She has presented one national level presentation and has some research and review articles. She is a life member of professional bodies like Maharashtra State Pharmacy Council and Indian Pharmaceutical Association. With this brief introduction, I now request ma'am to take over the session.
Thank you, Lou, for this introduction. Now, I'll start with my presentation so that we can start with the SCIENTIA session. Is my presentation visible? Okay. So my topic of today's presentation is malaria vaccine. Now there is a reason why I have taken up this topic is because to give you some information of how vaccines are designed, and the second is to make you aware that there is something called as malaria vaccine available for the administration to the infants and the children. So to begin with, you need to understand what is malaria and then what are vaccines. So we'll start with the malaria. You all must be knowing that malaria is caused by a protozoal parasite belonging to Plasmodium species. Anopheles mosquito is the carrier for it and is an infectious disease. Plasmodium species which are capable of malaria are Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malarian, Plasmodium novel. So out of this species, Plasmodium falciparum is considered to be the most fatal because it produces the symptoms which takes a long time of period to cure. Now we need to understand the life cycle of malaria. Female Anopheles mosquito are the carrier of this protozoa, Plasmodium species. So when this mosquito, which is carrying the protozoa, bite a healthy human being, the sporozoids get into the primary site where they infect is the liver. They get converted into schizons over here. Now there are two classes or two cycles in life cycle of malaria. The first is the asexual cycle, which happens in the blood, that is erythrocytic stage it is called as. The second is the sexual cycle, which happens in the gut of the mosquito. Once the schizons are being formed in the liver, they enter the blood, that is enter the erythrocytic stage. Now here from where the asexual cycle takes place. It gets converted into tropozoites and then to schizons, which ruptures and eventually forms merozoites. The presence of merozoites in the erythrocytic stage are the ones which are showing you symptoms for the malaria, for example, fever, chill, enlargement of spleen, etc. The tropozoites also have the capability to get into the sexual phase. That means they will get into stage 1, stage 2, and after that they'll differentiate into male gametocyte and female gametocyte. So now in the blood of the person infected by malaria have the gametocytes differentiated into male and female gametocytes. So when any other mosquito come and bite that individual suffering from malaria, those gametocytes get into the gut of the mosquito and where the fertilization takes place. So here we can conclude that a sexual cycle happens in the human body and the sexual cycle happens in the gut of the mosquito, forming into the gametocytes fused together to form ookinete, then cysts, then ookinites, then zygotes, and finally into sporozoites. These sporozoids get into the salivary gland. So this mosquito again becomes the carrier of the Plasmodium protozoa, and that mosquito, if bite any other healthy individual, will lead to spread of malaria. So this is the basic life cycle of malaria. Now, when you want to design any medicines, drug or vaccine or anything, you need to understand this life cycle first, and then you will target the particular stages where you can design a drug. Looking into the symptoms for malaria, you must all be aware of symptoms of malaria like fever, chills, fainting, enlargement of spleen, then nausea, vomiting, etc. Now we will take into consideration, like if you want to design a vaccine. For example, here we are talking into or looking into malaria vaccine. You need to do consideration about the following points. The first is the parasitic diversity. As I had shown you in the first slide, there were different Plasmodium species like Plasmodium falciparum, then you have vivax, oval, etc. So you need to understand that if you are designing a vaccine, whether it is specific to a particular Plasmodium species, or it is effective for all kind of Plasmodium species. So that is one thing which you need to consider. The second is whether it is addressing the source or the symptom. It depends upon which kind of immunity it is producing. For example, if it is producing antiparasitic immunity, it is addressing the source. If it is antitoxic immunity, it is addressing the symptoms. Now, this kind of immunity, as we are discussing vaccine, we need to understand this immunity in detail. First, talking about anti-parasitic immunity. As the name itself tells, parasitic, that means it is going to be very specific to the parasites. Now here, parasites are the source which cause malaria. So it addresses the source. The antibody response include humoral immunity and cell-mediated immunity. So ideally, a vaccine should enable the development of anti-plasmodium antibodies in addition to generating an elevated cell-mediated response. So this is about the anti-parasitic immunity. The next kind of immunity is antitoxic immunity. Now, antitoxic immunity is a immunity which is used to suppresses the symptoms. The symptoms, as we have seen in the previous slide, were fever, chill, enlargement of skin, body pain, nausea, vomiting. So all this kind of symptoms, mostly because of the merozoites present in the erythrocytic stage. Now, if our body is producing an immunity which is antitoxic immunity, it will suppress this kind of symptoms. So it reduces the effect of any toxic byproduct have on the development of the disease. So these were the two kind of immunity which a vaccine is responsible to develop. So how should be our ideal vaccine? It should generate cell-mediated and humoral antibody response, first thing. Second, it should increase the rate of parasite clearance. That means it should be able, if a person is infected by a parasite, then it should have the capability to clear out that parasite from the body. And the next is to reduce symptom and give future immunity. It should have the capability that even in the future that person comes in contact with such kind of Plasmodium species again, they should already have that innate immunity in them. Now, for any drug designing, we need to know the target. Now here, when you talk about designing a malarial vaccine, we have already seen the life cycle of malaria. Based on that, we can have target in three phases. The first is the hepatic phase, second is the blood phase, and the last is the sexual phase. So if a vaccine is effective in a hepatic state, that means the primary site where the sporozoites from the mosquito enters or affect and convert into merozoites through a sexual cycle, those type of vaccines will be called as pre-erythrocytic vaccine. If it is capable of acting on a blood phase, it will be called as blood stage vaccine. Now, if you have understood the life cycle of malaria, you must be knowing that in erythrocytic state, you will have the final product of the asexual cycle. Also, you will have the distinguished female and male gametocytes, which are responsible for the transmission of malaria to the other healthy individual. So this is very important phase or where the vaccine can be designed. The last is the sexual phase. Now, sexual phase happens in the gut of the mosquito. We cannot go and vaccinate the mosquito. So what we are going to do over here is we are going to design a vaccine such that the male and female gametocytes are not properly developed or are not developed, because of which the life cycle of the malaria is not being completed. And this is very important when you talk about the community welfare point of view. This kind of vaccines are called as transmission blocking vaccine. Next is hepatic phase. So we are going to see what are the agents developed in all this kind of stages, the three stages which we have seen in the previous slide. The first is CSP, which is called as circumsporocytes protein. Now here, the limitation of this kind of agent is it has low protective immunity, low efficacy, low reactogenicity. The first agent which has come up in the market is RTSS. It consists of Plasmodium falciparum circumsporozoid protein from the pre-erythrocytic stage. That means the stage before the erythrocytic stage. That means it is still in the liver. That is a hepatic stage. That's why this comes under the category of hepatic stage. The antigen causes the production of antibody capable of preventing the invasion of hepatocytes and additionally elicit a cellular response enabling the destruction of infected hepatocytes. There are some limitation to this kind of agent, and to overcome that, what they have done is they have put some chemical conjugates. So next, what they have done is we initially had Plasmodium falciparum circumsporozoids protein. Along with that, they also have put some chemical adjuvant and a surface antigen from hepatitis B. Three things together have resulted in the formation of RTSS-AS01. The brand name is Mosquirus, and this is the only malarial vaccine right now available in the market for the vaccination against malaria. The organization which has funded this vaccine are Malaria Vaccine Initiative, Bill and Melinda Gates Foundation, and JSK. Now, if we see the development of this RTSS, it has happened in November 2012, where it has entered the phase 3 study, and mostly it was found safe in young infant. In October 2013, JSK formulated malaria vaccine named RTSS. They have started naming it. Reduces the amount of cases among the young children by 50% and infant by 25%. GSK submitted an application for marketing license with the European Medicine Agency, that is called as EMA, in July 2014. On 24 July 2015, Mosquirus got approval from the EMA for vaccinating children with the age group of 6 weeks to 17 months. So this was a biggest achievement where the smaller age group were prevented from getting infected by malaria. WHO have formulated a policy on the use of this vaccine by the end of 2015 in the National Immunization Program. So this was the first agent in the hepatic stage, hepatic phase. The second one is NYCVAC PF7. It is a multi-stage vaccine. Like as we have seen the previous one, it was effective only in the pre-erythrocytic stage, that is hepatic stage. This is a multi-stage vaccine. Here you can see I had listed out the stages where this is effective. You can go through it. So we have completed the agent which are being used in the hepatic phase. Second phase comes the blood phase. Now blood phase, we'll go back and just revise what is happening in the blood phase. We all know that in the blood phase there is a asexual cycle happening, where at the end, because of the rupturing of the schizons and the formation of merozoites, the symptoms of malaria have been developed. Also, there is differentiation of the gametocytes, that is male and female gametocytes. So vaccine based on blood stage of the parasite can function as first as total elimination, or can cause a significant reduction in the parasite. So we can go in designing either by complete eradication or the reduction of the parasites. So antigen of blood stage for vaccine candidate are: first is apical membrane antigen, that one, that is AMA1. Then you have Plasmodium falciparum erythrocyte membrane protein 1, merozoite surface protein 1, and all these agents are in phase 2 clinical trial only. None of them have entered phase 3 yet. There was only one approved vaccine in the hepatic stage, and that has been approved by WHO for use. In blood stage, there is no such vaccine till date available for the use of the common individual. So here there is a combination B vaccine which consists of merozoite surface protein 1, 2, and also it contains RESA, which is ring-infected erythrocyte surface antigen. The combination of this is being named as combination B vaccine. 5 clinical trials have already been done and it is showing a promising result. Now, we all know that blood stage contain sexual stage and the sexual stage. Now, we are going to talk about sexual stage, which happened in the gut of the mosquito. It is very important for preventing the spread of malaria, because this is the only stage which will make the spread of malaria prominent. And if you have a vaccine at this stage, it will obviously lead to the prevention of the spread of malaria. This will not give any protection, though this you need to take care that if there is a vaccine at this sexual stage, it is not going to give you any protection or benefit to the individual who are already affected, but it is surely going to prevent the spread of malaria. The agent in the development are TBVs, which are called as transmission blocking vaccine. As the name itself tells you that transmission of the malaria is going to be stopped after administrating this kind of vaccine. So there is some unique biological advantage is low number of target parasite stage, a comparatively long vulnerable extracellular period, and lastly to reduce the antigen polymorphism, that means development of resistance. Now, efficacy of this type of vaccine can be evaluated in two stages, that is reducing the proportion of mosquito that becomes infected when fed through the membrane upon the gametocyte, that is when it is injected to a healthy individual, or it is injected to the one which is already been infected. Also, efficacy can be shown by reducing in incidence of the malarial infection. The next agent is SSM-VITM. SSM stands for sexual sporogenic mosquito stage. Now, this is the target where the vaccine to interrupt malarial transmission are going to act. So it is together named as SSM-VITM. This prevent the transmission of malaria from the infected person to the female mosquito rather than those involved in parasite infection of the human host or causing malaria disease. The vaccine will need to be amenable to campaign administration. So whenever we talk about any vaccine which has been administered or which is designed in a view for a campaign administration, following points you need to understand or you need to consider is the dose of this vaccine should be as small as possible, preferably a single dose. The second one is it should be effective for as long as possible and the cost should be low. So for this, the funding has been given by Bill and Melinda Gates Rollback Malaria Partnership and WHO. For every drug to come up in the market, whether it is a solid phase drug, liquid, or a vaccine, there are some regulations which are needed for it has to undergo some regulatory pathway for its approval. But in 2010, US FDA have given a waiver, and where they have stated that no legal barrier to prevent a vaccine, such as if the vaccine are designed in a way of SSM TBVs, that is transmission blocking vaccine, they need not to get any license. And because of that, many of the foundations have come up for the development of such kind of vaccines. Now, when we talk about fertilization, fertilization can be blocked either by pre-fertilization phase. So we are going to have two kind of phases over here, pre-fertilization phase and post-fertilization phase. When we talk about pre-fertilization phase, it is going to include agglutination, steric hindrance, complete mediated lysis of gametes, or else in post-fertilization phase, we are going to include complete mediated lysis, antibody-dependent phagocytosis, and blocking recognition of the mid-gut epithelial cell. For pre-fertilization, antigens are, you need to consider that antigens are the protein expressed on the surface of male and female gametocytes. Now, in the erythrocytic state, we have the development of male and female gametocytes. So, the proteins which are present on the surface of this kind of gametes can be taken as an antigen, and based on that, some modification done on the genetic coding, and based on that we can design a vaccine. So monoclonal antibody against either these proteins can block the infectivity of the gamete to the mosquito, and their blocking activity are enhanced by complement system. The agents are PFS, that is Plasmodium falciparum 4845. Now here the target, what we are doing is we are disrupting the gene, and preventing the fertilization to happen. When you talk about PS4845, there is PF230 also playing an important role in it. How is PFS 4845 contain a glycosyl phosphatidyl inositol, which act as an anchor that attach the protein to the surface of gamete, which will give you a platform, which will give the platform for soluble PF230 protein and allow the zygote formation. So this is the normal mechanism which happens. But if you have the surface of your PF4845 disrupted, the male gamete does not have the ability to get and attach to the female gamete. So here what you are doing is you are preventing the fertilization to happen. You are preventing the sexual cycle of the malaria to be blocked, and because of which the transmission or the spread of malaria is going to be affected. The next agent is HAP2. It has a conserved male protein. It is located on the surface of gamete plasma membrane, demonstrated to have a potent transmission blocking efficacy in both the rodent malaria parasite Plasmodium vagri and subsequently the human malaria parasite Plasmodium falciparum. Next, you have a post-fertilization. So this was about the pre-fertilization, where we have focused on doing some changes on male and female gametes. Here we are talking about post-fertilization. That means we will be focusing on the formation of zygotes or formation of oocytes or oocytes. Post-fertilization antigens are the protein expressed on the surface of zygotes and maturing oocytes from the parasite, form of the parasite. Anti-P25 antibody binds specifically to the surface of the parasites ranging from zygotes to oocytes. P28 is expressed slightly later in the development as anti-P28 antibody strains, mainly the retort and mature form of oocytes. Both of these are distributed evenly and abundantly over the entire oocyte surface, as seen by the immunofluorescent antibody staining and electron microscopy. There are 5 parasites-derived proteins which are currently acting as a lead for the designing of TBV, that is transmission blocking vaccine. These are the 3 given. We have already discussed about the P25, P28, about 48 and 45, which work in conjugation with P2230 is also been discussed, and we have also had a light on the HAP2. So this table will give you an idea about the current product development of the leading TBVs, and here you can go through it. This gives you the strength. This table will give you the strength and weaknesses. Now here again, this kind of this phase of vaccine are not be available in market. So these are the strength and weaknesses. We surely need to work on the weaknesses part and see that it successfully clear the phase 1 and different phases of clinical trial and come up in the market soon. These are the companies which are involved in the development of malarial vaccine. We have already told about the story of the development of the mosquito, where GSK is playing an important role. 3 foundations we have discussed, that is Bill and Melinda Gates Foundation, then you have the MVIs and GSK, which is playing a role in the development of the mosquitoes. Remember, mosquitoes is the only malaria vaccine till date available for the vaccinations of the children, and this is very important to note that because of this kind of vaccine, one day or the other, malaria, which is considered very fatal in some of the area of the world, for example South Africa, or where the in the weaker section of the society where the people are not able to take a proper drug for malaria, this kind of vaccines are going to play a very important role. Also, the TBVs, which we had talked about, that is transmission blocking vaccines, will also play a major role in preventing the spread of malaria. Remember, TBVs are not designed to prevent malaria. It is designed to prevent the spread of malaria. If the person is already infected by the malaria, the signs and symptoms won't be suppressed by such kind of vaccines. So here I'll put my conclusion to this presentation as the recent drug treatment for malaria is not that effective due to the development of resistance. We all know that many of the important diseases like tuberculosis, if we talk about cancer or we talk about malaria, most of the drugs have developed resistance, and because of which the oral availability of this kind of drug is to be overcome. So malarial vaccine is considered as a tool for eradication of malaria. WHO identified malarial vaccine as the most important cost-effective method to reduce the burden of this disease. And these are the references for the presentation. And thank you for your patient listening. I wind up my presentation over here. So any student, if have any doubt related to malaria vaccine, can put it in the chat box or can ask. So I think we should wind up this session now, as there is no questions from the other end.
Good afternoon, everyone. I would like to thank our distinguished guest, Mrs. Ekta Thakor, ma'am, for enlightening us with her knowledge. Today's webinar gave deep insights in the topic malarial vaccine. Ma'am focused on various points like from introduction to malaria to life cycle of malaria, signs and symptoms related to malaria, about malarial vaccine and antiparasitic immunity and antitoxic immunity in brief. She gave us idea about ideal vaccine, what are the potential targets, what are the blood phase, and what are combination vaccine. She also gave us idea about the companies involved in the development of malarial vaccine. Thank you once again, ma'am, and thank you all. We are looking forward to meeting you all in the upcoming session of SCIENTIA, which will be commenced at 2:30 p.m. and link would be same. Have a wonderful day ahead. Thank you all.